Hi, my name is Cheick Sissoko. I'm a biomedical engineering PhD student at the University of Cincinnati in Ohio, and I am the proud recipient of a graduate student fellowship from the IFER. Thank you so much for supporting animal free research. My project goal is to generate a human relevant neurovascular unit slash brain blood barrier on a microfluidic chip. And here is why. The brain is a privileged environment. In other words, it's so important that what goes in and what comes out is highly regulated. It's actually protected by a unique vasculature called the brain blood barrier. This barrier plays an essential role in brain homeostasis, but also protects the brain. And sometimes it does it too well because when we want to deliver therapy effectively within the brain, it's sometimes a challenge to circumvent the brain blood barrier. Interestingly enough, its dysfunction is correlated with several neurodegenerative disease and neurological conditions such as strokes. Instead of using animal experiment to study this barrier, I want to build a miniature human relevant microphysiological system that recreates some of the key features of the human blood brain barrier. I want to be able to create tiny perfusible microvessels, essentially be able to flow things like real blood. I want to be able to modulate the property of this barrier by optimizing the cell type that I include, by optimizing the gel, by optimizing some of the matrix. And I want to be able to integrate all of this with an IPS derived brain tissue like structure within a microfluidic chip so that the biology is purely human and not a proxy. Long term, my career goal is to help drive the adoption of non animal and human based research technology, not just as an alternative, but hopefully one day as a new standard for studying complex human biology. Thank you again for the IFER for supporting humane science and for helping make all this work possible.